Right. Thank you so much for the introductions and the invitations. I'm very excited today to talk about what we have been doing on sea-free plant genomics. So I think by now it's pretty obvious that thanks to the drops of sequencing costs, plant genomes are being published on a weekly or even monthly basis. And here's a list I grabbed from the list of sequence genomes I grabbed from this website. And while there are 300 or even 400 plant genomes out there, the vast majority of them were coming from seed plants. And very little are coming from seed-free plants. And so last year, we kind of summarized the current landscape of seed-free plant genomics in this figure. And so here's the seed plants, or the lower plants on this tree. And up here, we have the higher plants, which are the seed-free plants. And you have the bryophytes, lycophytes, and ferns. And you can see genomes available from this group of plants, but they are phylogenetically very restricted. So for example, if you look into the ferns, there are 2 genomes that were published at a time. They are coming from Salviniales, the same family, and the sister genera. And the same is true for the bryophytes. And again, we have multiple bryophyte genomes, but if you look at the liverworts, the homeworts, they all come from the same genus. So again, all the available genomes, they do not capture at all the whole phylogenetic diversity, and especially in liverworts and homeworts. So today I'm going to give you updates on what we have been doing on seed-free plant genomics. I will first talk about ferns, and I will introduce 2 new genomes that I've been working on: Alsophila, which is a tree fern, Marsilea, which is a heterosporous ferns. I will then switch to Lycophytes, focusing on Isoetes, and I will talk really briefly about diphasiastron genomes from Lycophyte. And finally, I will talk about homeworts, and we've been sequencing homeworts from each of the families. Okay, so first I will begin with ferns. Fern genomes are difficult. This is a very famous chromosome squash of a fern called Ophioglossum, and they have over 1,400 chromosomes. And ferns not only have really a lot of chromosomes, but they also have big genomes too. So this is Mesipteris. This is a fern. Its genome size is around 150 gigabases. And if you take all the known fern genome size, average them, you get around 12 gigabases. And this is significantly higher than seed plants. And here I put tomatoes and Arabidopsis, which is a fun comparison. You don't even see Arabidopsis here because it's very small in comparison. So a few years ago, when we were planning to get the fern genomics off the ground, we picked the lowest hanging fruits, and that is in Salviniales, or the heterosporous ferns. There are 2 families in Salviniales, Marsileaceae and Salviniaceae. And in general, they have much smaller genome sizes compared to the fern average. So they are from 0.25 to around 2 gigabases. And Salvina and Zola showing here, they have the smallest genome size. So we'll pick them to sequence. And after some work in 2018, we'll publish their genomes. There's our cool stuff we uncovered, including the evidence of whole genome duplications in ferns, a cool case of horizontal gene transfer of insecticidal genes from bacteria to ferns. Zola has symbiosis with cyanobacteria, and so we also look at their symbiotic interactions with the plants. We also did some transcriptome RNA experiments and to find some candidate genes might be involved in cyanobacteria symbiosis. Right, so the paper is published, and I don't want to go too much into details, but I want to highlight that Salvina and Zola, they are coming from the same family. It will be really nice if we can expand our sampling a bit further. And so with that, we're focusing on Marsilea, which is in Marsileaceae, the sister family. Well, now, I know it might not seem a big deal, to just add a sister family, but I want to point out that the divergence time between the 2 families are very deep. So around 187 million years ago, and this predates the divergence dicots and monocots. So even just adding a new family in terms of cutting the branches, this is quite important. So for Marsilea genomes, we're focusing on Marsilea vestita. So this particular species has been used by Stephen Woniak at the University of Maryland as a model species to study the rapid spermatogenesis. His group has developed a suite of genetic tools to study Marsilea vestita, and including the first time that RNAi was used in ferns. So a brief intro about spermatogenesis. In Marsilea, it's very rapid, from the rehydration of the microspores to a release of 16 sperms. This can be completed within 8 hours or so. And it was thought that this whole process does not require any new RNA transcriptions. And instead, it's believed that the microspores actually store a pool of pre-mRNA that is incompletely spliced. And after the rehydration of spores, this intron got spliced out and the translations happened. All right, so this process was described in this pretty seminal paper by Boothby et al. In 2013, and this was published shortly before Stephen Woniak retired from the University of Maryland. At the time, however, they did not have a genome. So instead, they compared the de novo RNA-seq, de novo transcriptome assemblies derived from 3 different time points, at 1.2, 3 to 5, 6 to 8 hours after the rehydration of spores. And they did de novo assemblies and compared the transcripts. So in total, they got over 180,000 transcripts, and of which 10,000 of them have introns. They also noted that the fully spliced transcript appeared later. So for example, they found this kind of transcript still have an intron hanging around early in the stage, and the fully spliced appeared only later. So this is consistent with the idea that they have unspliced introns early in the developments. Obviously, having a genome would be extremely helpful in precisely quantifying intron retention and splicing during spermatogenesis. Again, Stephen Woniak has since retired, but his colleague Steve Mount at Maryland still keeps Marsilea plants alive, and we started to collaborate on this genome. Li Guo, who is a former postdoc, did the nanopore sequence in the lab, and Nassim did assembly annotations. So we're able to anchor around 80 to 90% of the genomes under 20 pseudochromosomes using Hi-C. And after that, we did gap filling. The contig M50 is around 29 megabases. So very, very nice genomes, very contiguous genome. So having this really nice reference, we map the Boothby et al. RNA-seq data back to the genome. Again, a reminder, he generated 3 time points, RNA-seq on 3 time points, 1 to 2, 3 to 5, and 6 to 8 hours, each with 3 replicates. Okay. So when I use a tool called IR Finder, which quantifies the level of intron retentions. So IR stands for intron retentions. So an IR ratio is calculated as a number of reads that continues into the introns, like this one, divided by the intronic reads plus the splice reads. So in other words, an intron with the IR ratio of 0.5 means that this intron is retained in around 50% of the transcripts. So the higher the IR ratios, the more introns you retained. However, contrary to what was reported by Boothby et al., we find very little evidence actually for genome-wide intron retentions. The vast majority of the introns have very low IR ratio, much lower than 0.1. And in addition, if the retained intron is being spliced out during the spermatogenesis, we expect to see IR ratio to drop through time, right? Because the intron got spliced out. But we actually see the opposite. We see there's a marginal increase of IR ratio over time. So we'll then next look at a few of genes that Boothby et al. Highlighted, a total of 17 of them, for further validations using RT-PCR and RNAi. Well, again, we'll pull these genes out and we examine their read mapping patterns. So here are 4 of the 17 genes, but they all show pretty much the same patterns. You can see RNA reads all piling up on the exonic regions, and very few of them, or none, are mapped to the introns. So this patterns at all with what they reported, and In other words, we couldn't find any evidence of unspliced introns in Marsilea, and the same is true in all the time points. Another key findings from the Boothby et al. Paper is there's no new transcriptions in spermatogenesis, and they believe the stored pre-mRNA is the only source of transcripts. But our reanalysis of their data with our genome as a reference found there are actually a lot of genes that are upregulated during the course. And in addition, these upregulated genes are enriched in spermatogenesis-related GO terms, such as those, flagella, axons, membranes, projections, cell projections, and so on. So it's pretty clear that transcription do happen in spermatogenesis. So to summarize this part, we think the earlier transcriptome studies by Boothby et al. Might have misassembled a lot of the transcripts that might cause overpredictions of intron retain introns. And this is a huge bummer because this is what we originally thought, this is not what we expect to find. And also the biological significance of intron retention in plants has not been entirely clear. Other than we know they exist. And Marsilea spermatogenesis has been always cited as a really nice case saying, intron retention do play a role in plants. But now we know it's probably not real. So yeah. But this is the take home so far from our preliminary analysis on the Marsilea genomes. And we're working on more on this front, comparing it to other ferns and looking at the histories of whole genome duplication and so on. All right, so let's go back to the fern phylogeny. So now we add another star in Marsileaceae. But again, it's very narrow, right? So they're all coming from Heterosperous Salviniales. We're missing a huge chunk of fern diversity. So next, I'm going to talk about exciting new genome coming from the scaly tree fern family, Salviniales. So this is a collaboration with Chen-Zi Li's group in Chinese Academy of Forestry. So his group assembled a really nice genome of the tree fern called Alsophila spinulosa, using a combination of PacBio, Illumina, and Hi-C scaffolding. So the genome is around 6.2 gigabases, and they were able to sort it out into 69 pseudochromosomes. It's highly repetitive. 4.6 of them are repeats. So David Wickel, a graduate student at Cornell, and I were invited to join the team to, poke around the genome and see what kind of cool stuff we can find. One of the first things we did is to look into whole genome duplications, or WGD. So David used a combination of tools, the KS plots, gene tree species tree conciliations, and we find a very solid evidence that there's at least one round of whole genome duplications at the base of Salviniales. So, so here in the base of Salviniales. And this is at least 100 million years ago. What is really remarkable is that you can see the synteny got conserved extremely well, even though it happened over 100 million years ago. So this is quite striking, right? This is an old whole genome duplication events, and yet the synteny is still there. And this is again a stark contrast with what we find in Ceratopsia genomes. And Ceratopsia genome is forthcoming as well, is led by Blaine Marchant. So what they find in the Ceratopsia genomes is there's probably a whole genome duplication around 60 million years ago, give or take, but there's very little synteny left after that event. Whereas here in Alsophila tree ferns, they have a much older whole genome duplications, but yet the synteny has been kept really well. So we started to think, what's going on, right? What might be behind this really stark contrast? Could it be related to tree ferns being, tree ferns? It's quite well known that tree ferns have a much slower evolution rates, at least in the chloroplast genes, and which is associated with evolutions of arborescence. But it's unknown if the nuclear genes also have a slower rate as well. So of course, we look into this. David took a bunch of tree fern transcriptomes as well as some other all-group transcriptomes. He pulled out single copy genes and tested for nucleotide substitution rate shift. And indeed, we found most of the single copy genes that he looked at have decelerations when he enters the tree fern clades. This is very similar to what we saw in chloroplast genes. So in other words, the rate deceleration is ubiquitous, is genome-wide. So we therefore think that perhaps tree ferns are just really slow, both in terms of nucleotide substitutions and also genome rearrangements. And that's why we see such a remarkable conservations of synteny from such an ancient whole genome duplications. So I think it was maybe Mike Barker once in the botany after David presented these results, and he noticed that this is just like a salmon, right? Salmon, the fish. They also have a remarkable conservation of synteny and have a very slow rate of genome fractionations. All right, so there are a lot of other cool stuff about Alsophila genome I didn't get the chance to talk about, but the paper is accepted and should be coming out soon. And our collaborator did a lot more analysis on lignins, secondary metabolites, and so on. So keep an eye on the paper. All right, so that's about ferns, and let's move on to lycophytes. I will first talk about Isoetes, and I will give a really short teaser on Diphasiastrum. So before 2021, there have been 3 lycophyte genomes published, but they all come from Salmonella. And Salmonella is not the only exempt members of lycophytes, right? And there are 3 major lineages, Isoetes shown on top here, Salmonella, and Lycopodiales, and here's a Wupertia as a representative here. And they all have very different body plans and life histories. And in particular, we really like Isoetes because they are really cool. They are mostly aquatic. They have roots that are bifurcating and it's bizarre. And what really drew our attention is they have chem photosynthesis. So chem has evolved independently multiple times across land plants, right? But mostly as adaptation to dry environments to prevent water loss. But it was also involving Isoetes in aquatic environments, which is odd. So the key features of chem is that the carbon fixation took place during the nights and stored as malic acids. And during the day, CO2 is released, and then that enters the Calvin cycle and captured by rubisco. So it's a carbon concentrated mechanisms. And in areas, by temporarily separating carbon fixation processes, you can actually close the stomata during the day when it's the hottest outside and to prevent water loss. But in Isoetes, they live underwater almost all the time. So why do they need chem? And chem in Isoetes was first discovered by John Keeley in the 1980s in a species called Isoetes howellii. It's a vernal pool species. So it turned out vernal pool has a really unique CO2 variation profile. During the day, the dense vegetation in vernal pool carries out very active photosynthesis and depletes the available CO2 in the water. And when the night comes, CO2 level goes back up because of the respirations. So Isoetes howellii, instead of competing with the daytime CO2, they decided to do carbon fixation during the nights when there's the most CO2 in the water. And if you have nighttime CO2 fixations, then it pretty much qualifies you as a chem plant. But not every Isoetes species live in vernal pools. And for example, Isoetes bolanderi live in oligotrophic lake, which has very little vegetations, but it's also doing chem. And the explanation for this is there are just so little available CO2 in the water, and the surviving strategy is just fix CO2 all day long. And again, if you have nighttime CO2 fixations, then you behave like a chem plant. So Isoetes chem is an adaptation to CO2 limitations in aquatic environments. Again, they use this as a carbon concentrated mechanism. So consistent with this, Isoetes chem in many Isoetes is facultative. They're doing chem underwater, and doing C3 when the leaves are emergent and not limited by CO2 anymore. Again, terrestrial species, they tend to lose chem, and you cannot enforce them to do chem, even if you force them to grow underwater. So I think there are many flavors of chem in the genus. Some are obligate, some are facultative, some just don't have chem. And so it would be really cool to integrate physiology, ecology, and genetics to understand how chem has evolved in this really different lineage, and also across different habitats as well. And importantly, there are also quite a few chem genomes being published, for example, the Phalaenopsis orchids, pineapples, kalanchoes, but all these are flowering plants, right? So it will be really interesting throwing a Lycophyte plant in the mix and then see what's the level of convergent evolution we can see at the genomic and transcriptomic level. So to that end, we focus on Isoetes taiwanensis. There are a number of reasons. We know this species is diploids, which is very uncommon in Isoetes. Isoetes like to be polyploids that can go all the way to dodecaploids. So finding a diploid is very important. We have flow cytometry data, so we know the genome size, and we know they has chem. So this is a collaboration with Todd Michael at Salk. The UN did most of the DNA and RNA work, and David Wickel did all the data analysis. Right. So after a lot of work, we got a genome. We use a combination of technologies, just Illumina nanopore, as well as about a nano optical mapping. So once we have the genome, we started to explore its genome. And one of the first things we did is, again, looking at the histories of whole genome duplications. This is important because Salmonella actually there's no evidence of whole genome duplication in that entire lineage, which is really bizarre considering most of the other land plants have multiple rounds of whole genome duplication. So is this lack of whole genome duplication unique to Salmonella, or does that extend to Lycophytes, other Lycophytes as well? So again, we use multiple evidence, KS, synteny, gene tree species reconciliations, and we're able to find at least one whole genome duplications in Isoetes. So this is again suggests perhaps Salmonella is the outlier here, and Lycophytes having no WGD at all. Right. So the main goal of this project is to understand chem genetics in Isoetes. So we did a 30-hour time course. We sample tissues every 3 hours to track the gene expression profiles across throughout the day. And this is an ordination plot with all the samples and replicates plotted, and you can see them make a big circle and come back. And this is what we expect to find, we expect to see in a diurnal time course cycle. We also did a titratable acidity in parallel, just to make sure that we are it's doing chem. And again, the acid increased during the night and decreased during the day. It's classic chem patterns, chem cycling. So when I look at the expression profile of a few chem pathway genes in Isoetes and compare them with other chem plants, and namely pineapples, ananas, kalanchoes, and sedum. One gene in particular looks very interesting, and that is PAP-C. So shown here, basically catalyze the first carboxylation step, adding bicarbonate to PAP to form OAA. So there are two types of PAP-C. You can call them plant type and bacterial type. Plant type is involved in chem, in all the chem and C4 that we know of, although I know Carolina has a paper showing it might be involved, the bacterial type might also be involved in chem. And our finding is also consistent with what Carolina found in Yaka, that in Isoetes, the bacterial type actually has a much higher expression patterns and cycle stronger throughout the day compared to the plant type. So we don't quite know what bacterial type PAP-C in plants is doing, but so we couldn't really speculate much about its physiological or functional significance. But I think the recruitment of bacterial type PAP-C in Isoetes suggests that there might be more ways to evolve chem than previously recognized. Next, we'll look at the circadian clock regulations in Isoetes. So previous studies have shown that CAM and circadian clock are tightly linked. This makes sense because CAM rely on temporal separations of carbon fixations, right? And so the best way to achieve this is hook it up with circadian clock. But it seems that Isoetes has a really weird circadian regulations. So we look at a few classic circadian clock transcription factors. These transcription factors got turned on different specific time of day, which then initiate a bunch of downstream genes. So here, Isoetes are shown in blue, and Salviniales in orange, Arabidopsis in red. Some of the circadian clock transcription factors behave just like other plants, for example, LHY, but some like PR1, the Isoetes are antiphasic compared to other land plants, PR1 expression profiles. And in GI or Gigantea, it's typically a single copy gene across land plants. In Isoetes has two copies. One follows the typical expression pattern, the other one is antiphasic again. But then also look at the cis-regulatory elements. So these are the elements, motifs that the circadian clock transcription factors will bind. Again, things are weird. In particular, if you look at evening elements, or EE here, typically they should be enriched for genes that express during the night. But in Isoetes, is enriched for genes that express during the day. So all these suggest that circadian clock regulation in Isoetes is quite divergent, but whether or not that has something to do with CAM is unclear. A lot more aquatic plants would need to be done to know if this is specific to Isoetes CAM or not. But regardless, I think it's safe to say that Isoetes has a non-canonical pathway. All right, so that is what we have been doing on Isoetes. And I want to just highlight what we've been doing on the Lycophyte front. That's the only remaining lineage having no genome. So there are a number of genera in Lycophyte, and we don't have much genome size information for most of them, but for the ones we do, they are quite large in the gigabase range. And again, we picked the lowest hanging fruits that's in the Diphaseastron. So this is a joint project with JGI's Open Green Genomics, and they help us sequence the Diphaseastron genomes using PacBio, Illumina, and Hi-C. And we just recently got data back, assembly and annotation back, and it's looking really promising. Just right off the bat, we found very clear evidence of whole genome duplications based on the synteny, self-synteny plots. And our colleague, the Wang's group in Agricultural Genomics Institute at Shenzhen, also recently finished a chromosome genome assembly from a Hupertia species. And we're working together to compare our Diphaseastron genome with the Hupertia genome, which is much larger. And hopefully we'll get a joint publication out soon. All right, so that is what we've been doing on ferns and lycophytes. And now I'm going to move on to hornworts. So hornworts are one of the three bryophyte lineages, together with mosses and liverworts. And I think they are fabulous. They are really cool. First, they are phylogenetically important, meaning for the longest time, we have no idea where they sit on the plant tree of life. But the recent phylogenomic study seems to converge on this topology, that the bryophytes are monophyletic and the hornworts are sister to mosses plus liverworts. However, despite the monophyly relationship, I want to emphasize that the divergence time is very deep. So hornworts diverge from moss plus liverworts around 483 million years ago, and that's just very close to what hornworts and Arabidopsis diverged. So the hornworts are very deep lineage on the land plants. Oops, yeah. And hornworts also are weird in many aspects. They're sporophyte, showing here. They have a basal meristem instead of the apical meristem we see usually in other land plants. And the spores mature from the top and the whole thing got pushed out from the bottom. And we have done some analysis to see if some of the well-known meristem identity genes found in flowering plants could also involve in homeward sporophyte developments. And so showing here, hornworts have various reduced collections of transcription factors. They have one Knox, one Bell, and one Yabi, and one Leafy, and very few Wax genes. And some of them have much higher expression level in the sporophytes. So I think this is a good start, and it'll be really interesting to further characterize these genes in details and see to what extent the gene network overlap between the homeward basal meristem, shown here, and the apical meristem in other land plants. Another cool features of homewarts is they have stomata on the sporophytes. But in many aspects, they are very different from flowering plant stomata. There's no apparent active control of stomata. So the young gut cells will form at the base, and then they will open. And once they open, they remain open. They will never close. So the function of stomata in homeward has puzzled the botanists for ages. And the main hypothesis nowadays is the homeward stomata are for drying out spores to facilitate the dispersal and not really for gas exchange in the classic sense. But regardless of the functions, our preliminary data suggests that a lot of the key stomata pattern genes are upregulated in sporophytes compared to the gametophytes, like screen, speechless, too many mouth, and so on. Again, suggests that they might share some homology at the genetic level, but a lot more functional studies are needed. Homewarts also have sex chromosomes, and that's just wild. In this case, they're called a UV system, unlike XY, because the zygote and sporophytes are always heterozygous as UV. And Pete Schraffam, he's now an NSF plant genome postdoc in my group, and his main project is working on homeward sex chromosomes. Homewarts also have unique photoreceptor called neochrome. So it's a chimeric part of it coming from far-light sensing phytochrome. Part of them are coming from the right sensing holotropic. This is unique only in ferns and homewarts. So again, a very unique photoreceptor. And in homewarts, we have no idea what neochrome is doing, what kind of fixed functional rules they play. It's unclear. And lastly, and another thing that homewarts are weird is they have cyanobacteria symbiosis. And this is happening in all the homeward species that we know of. They will form these slime cavities, and they will attract the cyanobacteria, come into it, and the cyanobacteria will fix nitrogen for the plants. Plant provides sugars. It's a symbiotic interactions. Some of the homewarts also have a pyrenoid-based carbon concentrating mechanisms. And in a sense, this is like C4 and CHEM, but they do this in a single cell level, and they use a biophysical way to concentrate CO2. I will talk more about this later. So this is another really unique features about homewarts. So we published 3 homeward genomes from 2 Amphosphoris species, Amphosphoris agrestis and Amphosphoris punctatus, back in 2020. However, again, these are 2 closely related species, and there's no way they capture the whole genome diversity of homewarts. So to address this, we have been going out there and collect homewarts throughout the world and bring them back to a lab and to establish pure azenic cultures and sequence their genomes in-house using nanopore. So this is Jessica Nelson collecting homewarts in the cornfields. I don't know why. Some homewarts really like corns. And Pete Schraffam is now taking all the data together and assemble them into genomes. So right now we have genomes from every homeward families. So the ones in red are the ones we already have genomes from. Pretty high quality genomes because we use nanopore. So this makes homeward the best sample plant phylum ever. So I'm hoping to get a plant homeward pan genomes done. So the species in the black are the one we're targeting right now. But already the genomes we have now cover the whole tree of life really well, and they also represent diverse trait combinations. Some of them have pyrenoids, some of them are dioicous or monoicous, some of them have stomata, some of them don't. So this makes a really nice framework going forward to do comparative genomics. Pete Chevron showed this with me a few days ago, which is about the high C scaffolding on the homeward called Phymatoceros. And with some help from face genomics, he was able to get 4 big chromosomes and 1 tiny guy back. So 1, 2, 3, 4, and 1 tiny bit back. And this is quite consistent with our cytology. We know they have 4 big autosomes and 1 tiny beanie UV chromosomes. So this is very exciting for us. It's almost too good to be true, but he's now looking at what exactly is this, what kind of genes are inside, and is a U or V? It's another thing we need to figure out. So in addition to genome sequencing, we've been also working really hard getting homeward transformation system going for homewards. So this is a homewards expression GFP, and we have been pretty good success in putting genes into homewards, but we're still having trouble getting CRISPR knockout to work yet. But we are fairly confident we can get there sometime. So what we eventually want to do is build homewards as a model lineage to study diverse trait evolutions. And in particular, we're interested in the carbon concentrated mechanisms, the pyrenoid-based, and also cyanobacteria symbiosis. And I want to really briefly talk about what we've been doing on their CCM part. So carbon concentrated mechanisms. The whole purpose of a carbon concentrated mechanism is because Rubisco, the carbon fixing enzyme, has a lot of issues. So Rubisco couldn't distinguish CO2 and O2 well. And when Rubisco react with O2, then you got photosynthesis, which is really wasteful. And that's why carbon concentrated mechanism comes in. The idea is if you can concentrate CO2 around Rubisco, you could really boost photosynthesis efficiencies. Again, there are many types of CCM, like C4 and CHEM. They use biochemical way to concentrate CO2 by first fixing CO2 with Pep-C. But there are also a biophysical way to do this, and this has been found in cyanobacteria, algae, and homewards, of course. The idea of biophysical CCM is quite simple. You first aggregate Rubisco in a special compartment, and you pump CO2 or bicarbonate into it. And such compartment is called carboxysomes in cyanobacteria, or pyrenoids in eukaryotic algae and homewards. So Chlamydomonas, this is a green algae that has been traditionally used as a model species to study pyrenoid-based CCM. So I don't have a time to go over all the details about how pyrenoid works, CCM works in Chlamydomonas. I just want to highlight one particular protein called APIC1 here. So APIC1 is a disorder protein that link Rubisco together, and having an aggregate Rubisco is the first step of building a pyrenoid. So again, APIC1, a disorder protein, it's repetitive, so it has 5 repeat motifs, and each of the repeat motifs can bind to a Rubisco large subunits. What is really cool is that you can mix APIC1 proteins and Rubisco in a test tube, and you get a phase separation. So this is kind of a preliminary pyrenoids can be formed in the epidorf. So we couldn't find APIC1 homolog in the homeward genomes, and this is likely because these older proteins tend to have very little sequence conservations. So in order to find homewards versions of APIC1 or APIC1 analog, we turn to do a co-immunoprecipitation or co-IP. So again, the idea is we made our own anti-Rubisco antibodies. We bind it to a magnetic bead, and then we react this with homeward lysates. This is a liverworts, by the way. We couldn't find homewards on BioRender. Just so forgive us. And we react it with the homolog lysates, and we use a magnet to pull down the beads along with the antibodies, the Rubisco, and anything that can bind to Rubisco. So when I elute this, send it to mass spec for protein identifications. And then we use our genome proteome as a reference to see what kind of stuff we got out. Here's a list of enriched gold terns in our pull down samples. And you can see we got a lot of photosynthesis, chloroplast, phallicoid related terms. And this suggests that our pilot studies might have worked. We pull down Rubisco and other stuff. So we've been going through our list, and we have this thing we call hype1, we call the hyped pyrenoid element 1. It's a disorder protein, and it's highly enriched in the co-IP sample, in the co-IP in the pull-down samples, and highly expressed in homologs with pyrenoids, and expressed slightly less or very low in the in species without pyrenoids. You also have repetitive motifs. So we're quite hyped about this hype1, and we're hoping to do more experiments to see if it can actually bind to Rubisco, and it can help homolog to make pyrenoids, aggregated Rubisco. All right, so with that, to summarize today's talk, I hope you have a feel that the seed-free plants are awesome, fabulous, and we're beginning to feel many critical genomic gaps. And we're also doing what we call the genome-inspired studies to understand the obscure, but I think very interesting biology as well. And lastly, we're looking for postdocs to specifically work on homolog center vector symbiosis, or anything related to what I talked about today. So if you're interested, please contact me, and I'm more than happy to chat with you. And with that, I want to thank NSF for funding, the current and past lab members. They generated all the data, analyzed all the data I talked about today, and a lot of collaborators that make all the genomes happen. So yeah, thank you.